So turning over to Tobias Kind, I actually was very interested. Tobias is
Tobias.
from... Hi? Tobias, sorry, my mistake. A little nervous. Tobias is actually from Leipzig. And so I'm going, okay, and now he's at the University Davis. How did that work? From Leipzig bis Berlin bis Davis, okay. And it turns out that he was at the Max Planck Institute in Metabolomics, and the whole group came over to the UC Davis. And so I'm like, okay, from Berlin, the beautiful city, to Davis, a cow pasture. So I'm going to leave this to him to explain.
Yeah, thank you for your introduction. And UC Davis actually started out as a cow farm for UC Berkeley, so that's the short story. I want to thank Alex Allardyce. Where is he? Outside somewhere hiding. For inviting me, because he virtually threatened to remove all the ChemAxon licenses if I don't come. So I said, okay, I'm coming. And the background is, of course, we had a lot of good talks with him and exchange of ideas. So I'm very grateful to be here. And today I will talk about what is actually metabolomics. What are we doing? And I will give some real-life examples where we used actually ChemAxon tools for our research. And so where we are located as a metabolomics lab is down here. So the modern life sciences trees consists of genomics, transcriptomics, proteomics, and metabolomics. And once the genes are fully sequenced, you can determine mRNA expression. You can do protein or peptide profiling and also metabolite profiling. So we are looking at a number of, let's say, 1,000 compounds at a time and trying to identify sugars, amino acids, small fatty acids. So if you're coming from drug industry, you would usually look only for 10 targets after administration of a drug in the serum. We look for the whole range of metabolites, at least we try. And what we do with metabolomics profiling is we take temporal and spatial snapshots of the metabolic states of plants and humans and bacteria and possibly also viruses. And we're doing that with a range of mass spectrometry and NMR technologies. So we have, I think, 10 or 12 instruments right now. And because we started new, we are pretty well equipped actually with I think, all kinds of instruments which you can buy, well, at least all technologies you can buy on the mass spectrometry market. But although those machines run 24/7, we spend most of the time doing data evaluation. So we spend, I think, around 80% in front of the computer trying to figure out what are we actually measuring there. And so I have this divided into 7 approaches where we use the tools. And the first approach is we investigated was the data sharing in chemistry. And here I listed the tools we used for that: MView, MolConvert, MSketch, IUPAC naming, for instance. And the problem was that actually there are no repositories for spectra, mass spectra, and also there's no semantic annotation of research texts. And everybody knows the CAS database, Beilstein database. These are large commercial databases which did that in the past. But we concluded from our research, and we published that in PLOS ONE this year already, that there has to be a change in how chemists and especially also people who are involved in spectral acquisition publish their results. So because the good way would basically, if you publish a structure or reaction, why would you put it in a picture on a paper? You want to have it in a reaction database. So why not directly submit the reaction into a reaction database and keep it there? And the same for high resolution spectra. It doesn't make sense to make an optical character recognition on a high resolution NMR or mass spectrum. That doesn't work. So. And that is what we used or investigated. So we basically said, okay, for existing publication, the INCHI key and PubChem IDs are correct. smiles, we cannot use them because every vendor puts out different canonical smiles, and we should submit spectra directly to repositories, and that works in GeneBank. So before you publish, you have to put your annotation from your sequencing experiment into GeneBank, and then you can publish, not the other way around. And so this is something, we publish digital structures in spectra, we have a loss of data, we put it on a picture in an analog paper, and then later we develop tools which are working for OCR and text mining, which obviously have errors, and then we put everything into a large chemistry or spectral database, and that's the same or similar as if you have a cow, you create a hamburger, and then out of the hamburger you want to create a cow. And so that is, this so-called hamburger to cow algorithm. And maybe after talking to Steve from IBM, I could remove the wishful thinking, because he showed a very nice application how to do that. And I know also many other vendors are successful, and even ChemAxon, in converting names to structures, structures to names, or pictures to structures, and structures to pictures, maybe not that important. So, but it's still a Jurassic Park technology. So maybe we have it already for certain applications. So, and that is a big problem. So we suggested to go the direct way, putting structures and spectra into databases. And the ideas for ChemAxon we have actually here, for instance, can chemicalize, which is the ChemAxon name to structure converter, annotate my chemistry paper with INCHI keys. So can it pull out all the structures, create from the SD file all the INCHI keys, and I can send the INCHI keys to the web publication, and then Google can basically index all those structures. Or for instance, can MSketch code already the CML or the SMARTS code, reaction code, into the picture which I publish in a journal? Because if I publish online, there will be a SVG or maybe a PDF version, but it can contain already the reaction or the complete structure in the picture file attached as XML or a container. So this was one approach. The second approach, where we used also ChemAxon tools, was going from mass spectrometry to the molecular formula. That's what we usually do in mass spectrometry. We measure an accurate mass, and with that accurate mass, we want to generate elemental compositions, hopefully correct ones. So we used here a lot of MolConvert, all kinds of calculational tools. I mentioned the command line CXCalc tool, which contains all the property calculations from ChemAxon, and also Instant-JChem, very important. So we try to create elemental formulas with mass spectrometry, and then query all those different databases like PubChem, Dictionary Natural Products, Drug Bank, TSCA, for getting possible structures we identified. And the pre-existing idea was here, can we actually take those databases and create a set of scaffolds for natural products, so that we basically can lower our search space, because we don't look for virtual reaction databases or very large databases. We are only interested in natural products. So can we maybe use one of or those three or four tools to create natural product scaffold databases? So because they exist in drug companies, but they don't exist in the public domain, so which is still a problem. And we published this already back in 2007 in BMC Bioinformatics in a paper which deals with this approach that you have a spectrum, you create formulas. You have too many formulas, but you can take the isotopic pattern filter of your elemental composition and then say, okay, if my isotopic patterns don't match, I will exclude that hit. If they match, then I will have a possibly correct formula. I can take this formula and then query databases like Chemical Abstracts, PubChem, or any other database which has a web front end. So I can basically put a list of 100 elemental compositions or 1,000 elemental compositions, and then I can send them to any other database. So this works, for instance, in case of ChemSpider and PubChem, but not in case of CAS, for instance, because they don't allow that. So, and the second step would be to integrate more MS tandem mass spectra matching and also retention time filters. And one of the outcomes, which is, I think, the most interesting one, was that how many actually molecular formulas exist in a space below 2000 Dalton if you only consider the elements CHNSOP. And this number was not known before. So we have, if we only consider those most important elements, we have 8 billion compounds which are represented in that gray space. If we use our rules, our 7 rules, we are suddenly going into that red box, and that is really important for mass spectrometry because you don't want to operate on this 8 billion elemental space. If we look at PubChem at this time, it was only here. If we look at the 50,000 elemental compositions from the natural product space, not the isomers, only the elemental compositions, then we're on this little speck there. So, and we basically operate here, maybe there in this space. And we have to keep in mind, and everybody knows who is working in cheminformatics, that every elemental composition can explode to several billion possible isomers, and then we're not even talking about stereoisomers. So it maybe can explode to several billion stereoisomers again, and then from those stereoisomers you could also create tautomers if you want, or resonance forms, which are important for chemistry. So these were 2 approaches we already published, and the third approach is organic reactions in silico, and for that one we used MSketch and the reactor. And so because we have databases, we know where we know that those molecules exist in humans or in plants, and that is the KEC or database, which is the Kyoto enzyme database. They have a set of 16,000 primary and secondary metabolites which exist in, I think, around 2,000 species. And so what we actually can do is we can take the reactor, as we have seen before, and use it for reaction planning for metabolic profiling. So because what we do in our runs, we measure, as I said before, we measure small acids, alcohols, hydroxy acids, monosaccharides, sugars, free fatty acids, and sterols. But we have to convert them into volatile components, and in mass spectrometry you always do that with a so-called silylation step. So the compounds become volatile enough to be analyzed. And although we do that in the lab, we also can do that in silico or in the virtual space and already pre-calculate all our compounds we expect. So this was the approach, although we didn't use reactor directly for it, was, for instance, published in Plant Journal and other journals where we used metabolic profiling to figure out what is existing in plants. So, and the idea is, of course, that users could, for instance, share reaction libraries instead of ChemAxon always writes up reaction libraries. So that would be maybe good for universities. And they, I mean, that is a killer tool, if you want. I mean, that's a Uber tool. So, you know, in organic chemistry, people should basically only work with reactor in the first maybe 1-2 years, and then, you know, learn from that and play around with reactions. So what we do for our mass spectrometry step, we have a so-called methoxymation step, and that will protect aldehyde and keto groups, and it's actually used for opening ring sugars. So that is the idea behind, because you don't want to have closed ring sugars and open ring sugars in one measurement. And then we add a little bit MSTFA, which is the silylation product, and then we create those so-called TMS components. And you can see the spectra here. They're very similar. So we cannot distinguish them if we put them in a high-resolution mass spectrometer. But if we have a chromatography beforehand, a gas chromatography, we can separate the Z and E isomer. So, and this reaction is actually a drawing from reactor, and I use that also for submitting metabolite databases and performing methoxymations and silylations to create such virtual reactions in silico. And yeah, so our fourth approach where we, in order to figure out what we're actually measuring, need actually a library with all those silylated compounds, was that in the beginning, we had to create a mass spectral and retention library for doing that. And you can imagine you have a lot of different products. So we used here, again, the Sketcher, Instant-JChem, and also a range of the JChem API tools. And the topic was to develop a gas chromatography mass spectrometry libraries for metabolic profiling. And you can imagine if you take a serum samples, 20 microliters, you put it into a GCMS or a comprehensive GC times GC, two-dimensional GCMS, you can see between 5,000 and 10,000 peaks. And you can run that for a series of maybe 5 to 10,000 different samples during the range of a month or years. And the problem is you only identify maybe around 150 instead of those 10,000. So, and that is because the library is so small compared to the range of metabolites. So we have right now around 17,000 animal, human, plant samples from 55 different species and from 248 metabolic studies. So we can actually go there and say, oh, tell me what is in a fruit fly or what is different in a fruit fly compared to a tomato, or what is in an Arabidopsis but not in humans. So we can actually ask intelligent questions with that. And the ideas were here that in order to figure out how does our library look like, we actually need some statistical tools. For instance, PCA, PLS, principal component analysis, or partial least square, to basically show a nice picture of what is in our database, or maybe automated Venn diagrams for database overlap. This was actually not published yet, but it's in review right now, and that library is actually existing already since several years, but the publication will be out this year. So, and what we did with the ChemAxon APIs, we used, for instance, the smarts counter, and I really like that in ChemAxon they have a very well documented and also publicly available code resource. So you go on the web, you look for something, you can go on the forum or the ChemAxon website, and you already can pull out working code. And then you just need to put a little bit on the top and a little bit on the end, so that you say, oh, I can get these and that results, and you're free to go, even if you're not a programmer. So we use the API for so-called smarts matching. So we had a list of smarts to basically distinguish or figure out what kind of alkenes, amines, aldehydes, ketones, and so we have in our database. And although I think we got this the smarts table from a public open source source, and we used the ChemAxon API for basically creating this table of components. And in a second place, what we can do is, in order to figure out database overlaps, not only by the numbers but by visualization, we can take, for instance, these metabolites which are in the KEC database, in the metabolite database. We can detect substructures, and we can, for instance, create a 1024-bit fingerprint, or we can take any other molecular descriptor to do that, and we can compress this multidimensional space into 1D or 2D or 3D by using either PCA or an HCA, or basically just do a very simple Tanimoto comparison. And so what we did, we took this fingerprint space and compressed it in a 2D. And now the green dots you see, that is the official metabolite database. And our red, all the red dots, that is our library. And you can see whenever dots overlap, they are the same structure. So you can go there, you can click on that one, figure out the idea, go back to the database itself, and you will have the same structure. And I think that is pretty nice, and obviously people used it already before for just visualization of the diversity space. So, because if we would be only here, maybe in the range of alkanes, although I don't know if these are alkanes right now, that would mean our library is not very diverse, and it wouldn't be very useful. But we have a diverse database, so it is actually useful, and we used it already several times for metabolic profiling experiments. So these were things we already published or going to publish. So in our fifth approach, we try to predict retention times, because whenever you run a chromatography, you can not only measure the retention times and put them into a database, but you could also go the other way around by saying, I know my molecules, and now I predict all the retention times on the liquid chromatography or gas chromatography scale, and then I try to figure out, can I do that for compound identification? So we used the pKa logD and also all the possible other molecular descriptors we can create with the ChemAxon tools, and created a table, and then tried to model a retention model with that. And so the problem here was that although we have validation sets for pKa and logD values, at least the values I found on the website, where the number of compounds was very small. So I basically have to trust if ChemAxon tells me that the pKa and logD works very well, that is correct. And in-house, we don't have large pKa and logD databases. So if maybe users have such compound collections, they could donate it and keep it as a maybe as a validation space. Although obviously people would use it for for prediction instead of validation. So yeah, that's for users share pKa and logD data. And so the basic idea again is you have a retention time, so which is chromatogram goes over 45 minutes, or if you have a UPLC, it would go only for over 10 minute range. And then you have logP ranges. So here substances with a logP around 2, they will elute very early, and very lipophilic substances, they will be late eluters. And if we can use logP and logD filters or logD component predictions, we can take a whole set of components and can try to predict, for instance, retention time. So here we have the experimental retention time, and this is the predicted. And you already can see that the standard error for the development, we used 90 components with 48 for the development set and 32 for the prediction set. The error is pretty large, 3.7 minutes. So the only thing you could do is you could say, okay, my compound will elute between 5 and 10 minutes, between 10 and 15, 10 and 15, 20, but you could not say, oh, it will elute at around 25.2 or something like this. And the problem is, of course, we used an external data set from somebody else, that we just don't have enough components for liquid chromatography modeling or retention time prediction. So we needed at least 1,000 diverse components to develop a very good and very stable retention time prediction model, which also has a very high prediction power, not only tries to model a correlation coefficient of 1.0, which is easy doable with a neural network. You get 1.0, but your algorithm will not have a very good prediction power. So that is the most important. So here the diversity and also the number of compounds is certainly important. Our sixth approach is using those tools like Marvin, MSketch, Instant-JChem, and also the calculator plugins for students, basically in telling them that if you are working in metabolomics or mass spectrometry, you have to handle this molecular spectra, including not only mass spectrometry but also NMR, and the structures itself as a unity. Okay, the spectra do not exist independent from the structures, but many chemistry students really have a hard time to go beyond just drawing a molecule. Because they were not exposed to the tools. So I think that is very important to expose students to those tools, that they can generate stereoisomers, resonance species, use Instant-JChem for their own, like as a lab notebook, or use Reactor for creative thinking. And for the university, it's very easy because we get free licenses. So I think everybody should make use, or at least universities should make use of that. And those teaching slides are publicly available, and the publication itself is on the way. It's under review, so I didn't put it on here because reviewers still may decide it's not good enough, but I'm pretty sure it will be accepted. And so this is where we talk about those cheminformatics tools and how we can use them for teaching, and not only in a virtual environment, but really on virtual machines, for instance. And here I come to our last approach. For instance, lipid analysis, and we used for this approach, we used again MSketch, Instant-JChem, calculator, and reactor. And lipids are important because they're parts of all membranes. So everybody has lipids, phospholipids, triacylglycerides in the belly region, and the importance was set by an NIH grant of around 70 million over the range of last years, where they gave $70 million basically to the Lipid Maps Consortium. And you may know that the Craig Venter Institute here in San Diego, or around San Diego, also is heavily investing in lipid analysis for biofuel research. So there are lots of facilities which are involved in that. And one point is not only to grow microbes or algae, but also analyze what kind of lipids are in there. So, and we can do that with mass spectrometry, or LCMS and GCMS very good. And so what we did, we created the structure libraries and the structures we got from lipid maps, and tried to do something useful with them by basically taking the structure, creating virtual spectra, virtual in-silico mass spectra, and now taking those in-silico mass spectra and matching them against experimental spectra. Because with our mass spectrometers, we have already experimental spectra, but we don't know what those are. If we have an exhaustive set of all lipids, we can create those in-silico spectra and match them against them with a normal library search, a dot product search, and there we go, we have it identified. So, and this data was, for instance, presented at several conferences, and the manuscript will be submitted soon. You can also download tables from our website. And very short, in the last maybe minute or so, we used a robot which has a so-called nano electrospray chip with 400 nozzles, and for every new sample he would use a new nozzle. We connect it to a low resolution or high resolution mass spectrometers. We create mass spectra and tandem mass spectra. And in lipids it's very easy, because if they undergo MS fragmentation, they will lose either this SN2 or this SN1 group, and they also will lose the head group. And these are specific fragments. So if you have a database of maybe 1 million lipids, you can just fragment them with the reactor, for instance, by saying, please lose this side chain, and that one. Get the structures, convert them to accurate masses, and look up the accurate masses, for instance, in a real spectrum. So this is the way we, in principle, did it. So we used Instant-JChem for storing the lipid database of maybe 44,000 glycerolipids, 440,000 triacylglycerols, and actually even worst cases cardiolipins, which are important lipids in the heart, which is a database of around 32 million lipids. And for that case, Instant-JChem was not used, but we used JChem base. And then we have those and can send them to Reactor, or maybe import them into Excel and do some further calculations for those fragmentation predictions. And here I'm already at the end. So I showed you 6 or 7 concepts where we use the ChemAxon tools over a range of years. I think since we used them since 2004. And so structural elucidation really requires a deep interaction between structure handling and spectral handling. And most important, those tools need to be integrated with those public available small molecule databases like PubChem, ChemSpider, and KEGG. And people need to use those APIs to connect cheminformatics tools with those databases. And the the ChemAxon tools in our case are technology enablers, because only with them we can daily handle our structures. We can use them for metabolomics method development and also for development of new structure elucidation techniques. And here I am at the end. I want to thank all the members of the lab, our boss, principal investigator Oliver Fien, all the sponsors and industrial sponsors, NIH and NSF sponsors, and especially I want to thank all the ChemAxon guys for the help on the forum, with all those little bugging questions. They always are very kind to respond and are very quick to respond. And that's it. I'm at the end. Thank you.
Thank you very much. That was a very impressive talk. Do we have any questions?
I have just one very small comment, that you mentioned that reactor can be used to fragment molecules, but we also offer a tool dedicated for fragmenting molecules. We call it fragmenter. It is using a kind of similar technology as reactor, but also provides some statistical tools to analyze the fragments.
I think I used the reactor because it had a GUI. And usually I like GUI tools better because I'm coming from the Windows side than the command line tools. And Fragmenter doesn't have a GUI.
It doesn't.
Yeah,
it
that's
doesn't.
why
You're
I used
right.
the reactor. And I just fiddled around it. It worked quite well. But thank you.
Any other questions? I just have a quick question, especially regarding spectral data. It's kind of been a hodgepodge of different file formats. I'm wondering how you manage that. Is there any consensus toward a standard like animal for spectral data that you can use?
So for the NMR, let's say, society, it's pretty clear it's JChem, compressed JChem. And for mass spectrometry, for multidimensional data, the Proteomics Society is leading in this way, or multiple proteomics groups, and they have MZ data, MZ XML as their formats, but they're still fighting. And for mass spectrometry, they are pretty lazy. So, but in this case, it's NetCDF, as already somebody mentioned, as a tool for one-dimensional data, but it cannot handle multi-dimensional data very easy. But nobody wrote tools for it, and also JChem. But it's a big problem.
Yeah, I'm aware of that. I want to thank you again, and this is very impressive. And